Good day, everyone. I'm Dr. John Ward, Director of the Coalition for Global Hepatitis Elimination at the Task Force for Global Health. It's my pleasure to welcome you all to the latest version of our webinar series in collaboration with NIH, where we explore research that is advancing progress toward goals for hepatitis B and hepatitis C elimination in the United States and globally. Today, we're going to be looking at a new testing option for the United States point-of-care HCV testing. Next slide. Again, welcome. Just housekeeping, we really encourage you as you hear the presentations to submit questions to the Q&A function of Zoom, and we'll do our best to answer those all along the way and seek to pull out those as best we can as time is available at the end during our panel discussion. The webinar and all webinars in the NIH Coalition series are available on our website at globalhep.org. You'll see the videos in full, as well as those edited for the individual presentations, along with the slide sets and a summary to capture highlights from the presentations for concise reading of the content in the webinar. So it's a great resource, and I encourage you to go and see the variety of content that's available there. Next slide. I want to thank my organizing committee, particularly Dr. Betsy Reed-Canoli, who is with the National Cancer Institute, and as well as members from a variety of the institutes and offices at NIH, as well as my colleagues at CDC. And they put together the program of the webinars each year. And I want to thank them again for their assistance with that. Next slide. Again, we have about 17 webinars available from the NIH webinar series. They range from looking at HCV vaccine development, new research for functional cures for hepatitis B, looking at biomarkers for hepatocellular carcinoma, to looking at racial and ethnic and social determinants of health, and looking at particular risk populations such as persons who inject drugs. So please, again, go to globalhep.org to look at the content in the webinar series. Next slide. As I mentioned, we're going to be looking at a new testing option for hepatitis C in the United States. This option has been available in other countries. It's great to see it now arriving in the U.S., as we now have curative options for hepatitis C, which makes elimination a very feasible goal for the country. But getting people diagnosed and reliably linked to care is our biggest challenge, and this testing option provides a new opportunity to meet that challenge so that no one dies any longer of curable hepatitis C infection. Next slide. I'll now turn it over to my colleague, Dr. Nate Furukawa from CDC, to moderate the program. Nate?
Hi, John. Thanks for the introduction. I'm Nate Furukawa, the Senior Advisor for Hepatitis C Elimination in the Division of Viral Hepatitis, and I'll play host for today's webinar. So hello, and thank you to everyone who is able to take the time out of their busy day and join us. And welcome to those who are tuning in later to watch this as a recording. So I'm really excited about this webinar because we have this incredible story to tell you about how a small group of people, many of whom are presenting today, got together to bring this point-of-care HCV RNA test to the US. So, in an era where there's so much in the news cycle about catastrophes, scandals, breaches of trust, it can be hard for a good news story to break through. And that's encapsulated by the saying, if it bleeds, it leads. But we're here to tell you a success story that is part of a larger good news story that is totally flying under the radar. And we want to broadcast this great example of how government, academia, and the commercial sector all got together and deliver for the public. So who knows, maybe we can convince some folks in the media to cover this. And as some of you may know, this test, in fact, requires a pretty deep finger stick and a larger amount of blood to run the test. Remember, if it bleeds, it leads. So really, this should be a leading story. So let me look at today's program here. So We've got an amazing lineup of speakers here who played key roles in bringing the point-of-care test to market. And I'll briefly introduce each of these speakers in advance of their presentations, but I wanted to let you know that their full bios can be found on the Coalition for Global Hepatitis Elimination's website. So Francis Collins will provide some words to kick off this webinar. Then Bruce Tromberg from NIH will provide an overview of the Rapid Acceleration of Diagnostics Technology Initiative. That's the initiative that housed the Expert HCV project. Then Eric Lai from the Independent Test Assessment Program, or ITAP, will talk us through the ITAP journey that this test took. Pam Miller was also slated to present alongside Eric, but unfortunately couldn't make it today due to a family event. We'll miss her. Next, we'll be followed by Jennifer Rakeman Cagno from Cepheid. She'll be providing some comments on Cepheid's experience of going through the ITAP process. Ines Garcia from FDA was also slated to join us, but couldn't get timely approval to join this panel, and so joins us in spirit, if not online as part of the audience. I'll return and share how CDC has been working to shape implementation strategies around getting this tool out to the field. And then lastly, Andrew Aronsohn from the American Association for the Study of Liver Diseases will come and describe the efforts to simplify Hep C treatment. So next slide. So unfortunately, Dr. Collins was not able to join us today due to an immovable conflict that arose, but he was able to record a voice memo for us, which we'll play shortly. And in some sense, our champion, Dr. Collins, doesn't need an introduction to this audience, but nonetheless, Dr. Collins is the former head of the National Institutes of Health and the current lead of the National Plan to Eliminate Hepatitis C. During his time in the White House as senior advisor to the president, he concocted this brilliant plan to eliminate hepatitis C, of which point-of-care testing was a crucial component. And he's brought together some incredible star power to hepatitis C that has made all of what follows possible. So without further ado, let's advance to the next slide and play the clip.
Hello, everyone. I'm Francis Collins, currently serving as the lead for the White House initiative aiming to eliminate hepatitis C in the United States. And I'm glad to join this webinar, although I'm sorry I'm doing it by a recorded video because I will be on an airplane at the time of the actual event. But very glad to have a chance to say a few words about this really remarkable national initiative that has the promise of saving many lives and many billions of dollars. What's not to love about that? Hepatitis C affects an estimated 4 million Americans. That's CDC's most recent estimate. We don't really know for sure because a lot of them don't know they have it. It has this long, slow course without much in the way of symptoms until you turn up with liver failure, liver cirrhosis, liver cancer, and sadly, often early death. But we have a cure. We've had a cure now for almost 10 years. But unfortunately, access to that cure is not available to many of those 4 million people. That doesn't seem right, and that has sparked our drive to action. When I was serving as the president's acting science advisor back in 2022, my senior advisor, Dr. Rachel Florence, and I identified hepatitis C as one of those areas that a full effort across the government could actually do something pretty remarkable. And with lots of assistance and advice from CDC and FDA, CMS, HRSA, SAMHSA, Indian Health Service, the Bureau of Prisons, we were able to outline a plan that ought to make it possible to reach out to most of those folks in a way that would be affordable and achievable in a 5-year period. So this led to the development of a bold program, which is currently the subject of bipartisan legislation sponsored by Senators Cassidy and Van Hollen, and which we believe has a reasonable chance, maybe even a pretty good chance, of being enacted into law. The 3 parts of the national initiative: one was a subscription model following the lead of a pilot in Louisiana to try to make the access to the cure affordable. The second was a comprehensive public health program of healthcare delivery outreach for prevention, for education, and care so that many of these marginalized populations could in fact be reached and successfully treated. But the third part, which we're talking about today, was the need for an acceleration of diagnostics to improve testing across the nation. Because At the time that we started this, somewhat shockingly, the US lacked a point of care test, even though other countries like Australia had those available for some time. In the US, it required oftentimes as many as 3 clinic visits before a diagnosis could be made and treatment could be started. And we know we lost a lot of people along the way because they weren't feeling ill and they had other things that they had to wrestle with. So point of care test allowing test and treat in a single visit became a big priority. We convened key federal agencies in the summer of 2022 to build support for this and proposed leveraging the success of the RADx ITAP program, which had fast-tracked COVID-19 home tests to considerable success, to do the same for hepatitis C. And in fact, with a lot of support, some tin cupping that I did across CDC and NIH and other folks who came forward to say, this is so important, let's figure out how to do it even before we have the legislation. The program rapidly progressed. And in somewhat of a record period of time, by June 24, FDA authorized a hepatitis C point-of-care diagnostic test based on rigorous clinical testing in the US, showing that it had the desired specificity and sensitivity. This was a significant milestone in this national effort. Buoyed by that success, RADx ITAP is now tackling another critical gap, the development of a test for hepatitis B, which will be a crucial step forward in managing both hepatitis epidemics in the US. So here we are in October of 2024, looking at the possibility of this program actually coming into being in the not too distant future, if in fact Congress can see their way forward to bipartisan support. And we're encouraged by the fact that the Congressional Budget Office has been signaling signs that they might very well see that this could be a program that actually saves money in the first 10 years because of all the healthcare problems that we will be able to prevent by getting people cured instead of having them afflicted downstream of various problems such as liver failure, liver cancer, and oh, by the way, also diabetes and kidney disease, which we know are part of this disease and which with cure can have their risk greatly reduced. So it's an exciting time, an exciting topic. I want to thank Nate for having put together such a great lineup of people to present, and happy to have a chance to kick this off and wish you all a really interesting conversation about this topic of how we're bringing the best of diagnostics forward to make this happen. Finally, I just want to say from my small but mighty team of Rachel Florence, who I've mentioned, Josh Sharfstein from Hopkins, Risha Irvin from Hopkins, and Kevin Klinkenberg from HHS, we are delighted to see that in this case, all of government coming together to do something that is going to be a great gift to humanity. Thank you all, and have a wonderful webinar. Hello, everyone.
Great. All right. So without further ado, our next speaker is Bruce Tromberg. Bruce serves as the director of the National Institute of Biomedical Imaging and Bioengineering at NIH. And in his role, Dr. Tromberg oversees research programs focused on developing, translating, and commercializing engineering, physical science, and computational technologies in biology and medicine. And this includes the Rapid Acceleration of Diagnostic Technology initiative, which I'll turn it over to him to tell you more about.
Thank you, Nate, and thanks to the coalition for putting this on. It's a really wonderful opportunity. Nate asked me to give a little background on how all these kinds of programs that we're using got started. So I'll go into RADx Tech, where it all came from, and how it couples into the Hep C initiative. Next slide, please. So this goes back to the heart of the pandemic in April of 2020, when Senator Alexander and Senator Blunt actually reached out to Dr. Collins saying, you know, we're not sure there's enough investment and capacity for diagnostics for COVID, and we're really interested in understanding what NIH is doing about this. And Dr. Collins was really well aware of initiatives that we at NIBIB had initiated in order to really go beyond laboratory testing and try to dramatically expand test platform type and performance so they could reach people in their homes or at the point of care. So this led to a conversation between Dr. Collins. He called me into the phone call. I explained a little bit about what our plans were to create a competitive innovation funnel type of approach that we could get innovators from all around the world to submit ideas and have a systematic way of evaluating them in what we called an innovation funnel. Senator Alexander wasn't quite understanding that well what I was talking about. And Francis said, you know, it's like a Shark Tank. And they immediately really grasped the concept, I suppose because of its popularity in sort of popular media circles, but it really kind of stuck. And just a few days after our conversation, we saw this editorial piece in the Washington Post by Senators Alexander and Blunt. We need more COVID tests, and we propose a Shark Tank to get us there. And that really helped drive and popularize the underlying idea behind what we were trying to do. We didn't know at the time, this was in sort of mid-April, the conversation, but on April 24th of 2020, there was a fourth congressional supplement, which included $1.5 billion in it to NIH for advancing testing platforms and technologies and actually distributing them to underserved and underrepresented populations all around the country. So that was the birth of the RADx programs, the TEC and the UP, the RADx underserved population programs. And our goal at the time was to try and expand point of care and over-the-counter testing in a very ambitious way so it would exceed ultimately laboratory testing. And we needed tens of millions of tests to be produced by fall of 2020, as articulated by these leaders. So we used this innovation funnel approach, and next slide, please. And we couldn't just launch a complete initiative from nothing. So we were able to build on a very well-established network that we at NIBIB had invested in, the Point of Care Technology Research Network, or POCTERN. And that involves a consortium of academic sites and industry, Johns Hopkins, UMass, Northwestern, Georgia Tech, Emory, CIMMYT, the Innovation Center at Mass General Hospital. And we added unique structures to this network so they could validate new technologies and ideas as they came across the transom. That was a validation core at Emory, Georgia Tech, a clinical studies core using standard design and digital health platforms, single IRB center network types of approaches so that we could evaluate test performance very quickly, as well as a deployment core dealing with supply chain, manufacturing, building user communities. And our primary vehicle for all of this consortium, which added many members of government, academia, and industry, swelling to nearly 1,000 participants early in the pandemic, was the innovation funnel, or the Shark Tank, where we received over 1,000 applications to come in over 3 different openings of that innovation funnel. Next slide, please. Well, at the beginning, I kind of thought this was a technical problem for us to solve. All we had to do was bring out all this wonderful innovation in technology and science and get them developed, and it would find its way to the public. But of course, there are many cultural and administrative and organizational hurdles to overcome. So the RADx program spawned off several other elements to the program, including RADx Mars, which is a digital health platform that allows mobile devices to be used for online fulfillment and delivery of tests to people's homes. We launched a program called Say Yes COVID Test that evaluated the idea of people testing in their homes in the first place. It had never really been done at scale. The largest scale had been in pregnancy tests, which is about 8 million a year. In February of 2022, we saw 1 billion tests being distributed to people in their homes. So we needed a major paradigm shift and need to evaluate whether or not this could occur. We launched a variant task force to deal with testing performance in the presence of rapidly changing variants. The really quite well known now, the ITAP program, the Independent Test Assessment Program, which deepened our relationship with FDA and allowed us to move through authorization at an unprecedented speed, which directly impacted the Hep C project, home test to treat programs, redesign of tests for more accessible So we launched all of these after that initial innovation funnel to solve many of the other problems, and we continue to maintain these structures. Next slide. So what's the bottom line impact? In terms of what our investments did for increasing test capacity over a 3-year period between April 2020 and April 2023, test capacity, we were able to increase by about 8 billion tests, 56 EUAs, 15 or actually a few more than that, multiplex COVID flu tests, including a recent de novo one that now has been authorized, and help drive this paradigm shift out of laboratories and into over-the-counter and point-of-care tests, which, next slide, please, have been driven through this incredible cooperation between government, industry, academia, all coming together around this ITAP process. So the FDA was incredibly enthusiastic about the potential for ITAP from the very top, from Jeff Shuren, the center director, the entire OHT7 team, the technical team, fully on board in helping us work with them to optimize workflows and improve accuracy, precision, and performance of tests as they came about. I won't go into details on this, but I think the really big feature to it is compressing the timeline through economization of multiple steps all along the way, ensuring that data are acquired independently. And Eric, Lai, and Pam perhaps may be able to comment on some of the details of that just after my talk. An important feature of this is we were able to provide the FDA with really large-scale quantitative evidence that helped inspire changes of FDA policy. One notable example is the idea of serial testing, which they embraced very strongly with the evidence that we were able to provide from these kinds of studies. Next slide. So what's next for RADx Tech? You've heard hopes for Hep B, which is an ongoing project. So in infectious disease, we've expanded, had the Hep C successes, but are also working hard to do STIs, HIV viral loads at point of care in regions where they are accessible, and have many different budding collaborations in infectious disease with lots of partners. So we've reached out. It's not any longer just a COVID-driven phenomenon. We form partnerships with multiple stakeholders across the NIH and other government agencies to try to advance and accelerate urgent needs, both in infectious disease as well as chronic disease and prevention. And we note neurotechnologies, maternal health technologies, fetal monitoring, endometriosis detection. These are all being done using the basic RADx framework of acceleration, working with regulatory and de-risking technologies so they really can find a truth application very, very quickly and move out to the public. Next slide. So to summarize, we've seen this experience driven by the pandemic, where consumers and people at home and the point of care are really a new diagnostic market at a scale that was really unanticipated and unprecedented prior to the pandemic. The opportunities here are to continue to keep this active so we can leverage a lot of these advances to be pandemic ready. We built digital health infrastructure, diagnostic technology infrastructure, and they need to be continued to be invested in for other challenges and problems. How do we get this into medicine is the next big challenge. Integration into healthcare, electronic health records, reimbursement, connecting the testing with sustainable economic systems. And then ultimately, this big driver of telehealth and telemedicine, which is looking at patients across a screen, but could benefit dramatically from technologies that could be easily deployed in people's homes and at their point of care to optimize clinical decision-making would be a wonderful coming together, illustrating the power of this approach. So with that, I'll finish.
Thank you, Bruce. Really appreciate that presentation. Our next speaker is Dr. Eric Lai, who previously served as the Senior Vice President and Head of Genomic Medicine at Takeda Pharmaceuticals before his retirement in 2018. But the pandemic brought Dr. Lai out of retirement, and frankly, we're not letting him going back. But he is rocking it as the team scientific and clinical lead for the independent test assessment program. So over to him to let him tell you more about the expert HCV ITAP journey.
Thank you, Nate. First of all, I have to apologize that my computer keep telling me that my internet is unstable. I actually miss most of Dr. Collins' video because of that. I am going to turn off my video so that hopefully my audio will still hold. So I was supposed to do this presentation with Pam Miller, who is the ITAP program manager, but she has a last-minute family conflict. So I'm going to do the presentation. Move. Next slide, please. Thank you. So Dr. Tromberg review briefly the ITAP process, and in this slide, we are showing the ITAP process, how it is applied to the HCV program. Now, the critical component of the ITAP process are shown in this slide. The technology de-risking phase, which is shown at the top, by the Emory University and the Grady Hospital test validation team was really critical to the planning and success of the project. The validation team confirmed the LOD very quickly of the assay that we were going to use, and the Grady Hospital performed a clinical study in a very short time. In a month, they were able to enroll over 100 subjects, so that we have very good confidence of the performance of the test and provided critical information on how to prepare for the clinical trial. In addition, the Emory testing lab also provided the fingerstick analytical data for the actual FDA submission. Now, that's a first for ITAP due to the complexity of the project and also the IRB limitation on some of the testing procedures that cannot be performed by CRO. Now, the urgency and the speed of the project were listed at the bottom of the slide. It took us 4 months with 15 sites to enroll 1,200 subjects. That is typically done similar to a phase 3 trial of a drug trial, and usually takes about a year. That was a lot of coordination. And the submission of the de novo from April 15 and get market authorization in June 29 took 73 days. It is really a process that usually takes easily 6 months. And I have to give a shout out to Wei Jiang, the regulatory manager at Cepheid, who was masterful in organizing the submission. As a result of all of the work of everybody, we completed the project under budget and 1 day ahead of schedule. Next slide, please. So what were some of the key success factors of this project? We have outlined it in this slide, and we highlighted some of the important points up in the top, especially applied to the HCV project. First, it was very clearly right from the top the visions of all of the leaders from the different agencies, all the way from Dr. Collins to Dr. Tromberg and CDC and FDA. Everybody was right on board in the beginning, and everybody had the same vision. And then the project team was empowered to accomplish the goal set up by the leadership. At no time did the leadership involve themselves with the operational aspect of the project team. They basically set the vision and say, go ahead and do it. Now, that's great. We have a clear lines of decision-making and authorization, and it will share the urgency around all the stakeholders that we get involved. To give you an example of the hands-off operation from the leaders, I remember at the second monthly update, when we reported that we had 5 positive subjects at month 2 of the clinical trial, and we need 120 for the whole trial. And Dr. Collins asked, how are we going to achieve the goal when we have almost 50% of the timeline of the clinical trial already gone by? Well, we did not let him see us sweat. But on the other hand, he also trusts us to carry out a plan that we assure him that we will be able to carry out. The important part is that we apply a proven process that we have carried out and performed with COVID, with the COVID flu multiplex, with monkeypox, and at that time, we had over 20 EUA and projects authorized. The other thing that's important is that we have selected very carefully the sponsor for the project. And we have a very good comfort level that we will have a high probability of success. And we engage all of the teams that are highly specialized in what they do. And our collaboration with the FDA, as stated by Dr. Tromberg, was absolutely critical. And all of the government agencies help us when we need help. Next slide, please. Now, what were some of the key challenges with this project? The first one I mentioned is that we had to manage a large number of clinical sites over a 4-month period. Usually, this would have taken us easily 12 months or more. That was a major work. And the other challenge was that there was no 510(k) clear sample collection device for fingerstick. So we need to develop a lot of training material for that blood collection process in order to make it happen. We also have difficulties in procuring some of these materials because there was a lot of materials, and a lot of them, it's all over, not just in the United States, but in a different part of the world. The timing and communication across many of the teams were also a challenge because in the US, we have people from the East Coast all the way to Hawaii. And then we have the Swedish group in Cepheid and South Africa. So any meetings that we have, somebody is going to be up either at 3 o'clock in the morning or going to bed at 3 o'clock in the morning. But we did it. We made it happen. We also have very, very fast identification of the clinical site, which I will mention in the next slide. And we have been able to do all of this in a very short time. So fast that some of our key challenges was actually in documenting some of the data and documentation issue often show up on the clinical path. And this is something that we have learned for our next project. Next slide, please. Now, communication, as I said, is really critical. We decided to divide our communication team so that it's clearly technical leadership execution. And we assign tasks to specific individuals, not groups. And we have very efficient hands-off. So, for example, dealing with the FDA, we have a minimum of twice a week calls with them, and I will have emails with them almost every day. And then every Friday at 3:15 in the afternoon, I will have a discussion phone call with Ennis in the FDA, and we go through the whole week as far as what we have gone through, what we need to do next week, and so forth. The collaboration and the handout with the FDA was absolutely critical with all of the all aspect of the project and throughout the whole project. And at the end, we all say that it is the people that makes it work, not the technology. Next slide, please. Now, there are some things that we have done with the HCV project that we have never done before in a prior ITAP program. The first one is the groundwork for identifying all of the clinical sites. We did extensive surveys. We engage networks that have a lot of information on the HCV principal investigators. We went into literature. We talked to the CRO so that we very quickly identify a lot of the clinical sites that were suitable for the HCV project. And luckily for us that Dr. Jennifer Haven at Kentucky have done a study using the device that subsequently ended up to being the de novo. Very big study that we can learn a lot of the information from. Now, we also, based on my former pharma training, realized that because of the size and the speed of the trial, we really have to have a dedicated clinical trial oversight team and person within ITAP to facilitate all of that work in the clinical trial. And that was something that we've never done before. And the last part, as I said before, Emory actually contribute significantly to the analytical data for the submission. And that is also something that we've never done before. Next slide, please. Now, what are some of the lessons learned? The first one I have to say is that we try really, really hard to control the scope creep. Now, during the whole project, there were a lot of wouldn't be nice to have. The problem is that every nice to have would translate into 1 to 2 months of additional work. Now, that would be fine for a project that lasts 18 to 24 months because you can try to catch up, but not for this project. So we try really, really hard to control that. The other thing is that while we thought that a CE mark product would be easier to adapt to the US, there was still significant adaptation and de-risking needed. And the third point was actually a really surprise for us. We thought that the blood collection would be challenging for a lot of people that inject drugs because they might have callous fingers for finger sticks. It turns out that the venous blood drawn in these patients, in these subjects, were actually more difficult than having finger sticks. We have more people that we end up not be able to get a venous blood drawn than the finger stick. So this showed the importance of the point of care finger stick assays that we have developed. Finally, a lot of the long lead time for acquiring some of these items from overseas require constant pushing from multiple agencies. And that is something that we have learned, and we are going to apply our learning to the HBV projects. Next slide, please. Now, with such a fun project in such a short time, we work very well, but then also there are times that we have interesting a directive from the different people that runs the project. I remember that when we first started doing the project, we tried really hard to have it as quickly as possible. And at the first meeting with Dr. Collins, we told him that we can do it in 12 months. And the first thing that came back to us was, can you do it 3 months faster? And that's why we end up at the end of June. And when we talked to the FDA, Uwe was very, very sure. And then he said, when in doubt, escalate it to us. And when we were doing our final submission, and we were working really hard on getting the de novo through by the end of June, there were discussion about, well, can we push it into July? And Ines from the FDA was very clear. There's no July. We have to get this done in June. And finally, because of all of the different parties involved and all the different reagents that we have to make sure that we ship to different parts and so forth, we actually have some of the reagents shipped to people's home over the weekend in order not to lose the weekend. And they were always saying that, can we please confirm that we are shipping this correctly? So those are some of the fun things that we learn in the project. Next slide, please. Now, at the end, it really takes a village to get this done. And a lot of people support this project. Just like a film credit, everyone listed here play a major role in the success of this project. And yes, there were more FDA folks on the list than CDC, NIH, and ITAP. And that is correct. FDA really put in a tremendous effort in this project. And I can tell you, every single one of them contribute significantly to the success of this project. And with that, I close and give it back to you, Nate. Thank you.
Thanks, Eric, for giving us a sneak peek of what it was like to get this across the finish line. Our next speaker is Dr. Jennifer Rakeman-Cagno, who is the Senior Director for Medical and Scientific Affairs under Public Health Programs at Cepheid. And in her role, she is focused on advancing diagnostics for infectious diseases of public health importance and pandemic preparedness. So I'm going to turn it over to her to share Cepheid's perspective on the expert HCV journey and what it was like going through ITAP.
Great. Thank you so much, Nate. And thank you so much for having us as part of this amazing webinar today. So we can go to the next slide. And as I talk about and thought about the Cepheid journey here, I really wanted to start with Cepheid's mission, which is delivering a better way to improve patient outcomes by enabling access to PCR testing everywhere. And the reason I wanted to reference this is because this really does embody the goals of this project in really ensuring that we could get this test that, as others have mentioned, had been available outside the US, getting it available in the US to patients who need it where they are. And so if we go to the next slide, I just want to talk a little bit about the history and the background as we came into this project. So as Dr. Collins alluded to, Cepheid had an HCV viral load fingerstick-based test available outside the US since about 2018, and had been commercialized across Europe, Africa, and Australia. And so in that sort of 5-year period before we thought about this project, we had learned a whole lot. And the test had a very big impact in these other locations outside the US. And what we learned was that if a site had access to the instrument and the test, was able to prescribe treatment, and had drug available, that really enabled this test to treat paradigm where patients could come in or be met where they are, get a test, get a diagnosis, and get linked to care and linked to treatment within a visit. And we sort of obviated those issues that resulted in loss to follow-up. We also learned that phlebotomy is challenging. Eric alluded to this as well, not only because it's sometimes challenging in patients, but also because it's challenging to do in very decentralized settings outside of a clinical outside of a clinical site. So capillary blood is really helpful because a finger stick collection can be done by a community health worker or a non-phlebotomy trained person and can be done in these decentralized settings. So if we go to the next slide, this just shows one of my favorite projects outside the US, which is the Combi Clinic in Australia. So in this case, the team works out of a 1975 Volkswagen bus. They have gene expert instruments on the bus, and they take it out to where patients who need HCV testing are. They meet them where they are. They do the testing on the bus. They get the result back to the patient and get the patient linked to treatment. So they walk away from that bus, if positive, with their first days of medication so that they can move towards a cure. So we can go to the next slide. So kind of where we started and the level of collaboration that happened in this project was just absolutely incredible. So as mentioned in early 2023, we had a conversation with the Federal Task Force, Dr. Collins and team from the White House, about this interest to bring this finger stick test that was available outside the US to the US to really focus on elimination programs in the US. Throughout the project, there was an incredible level of collaboration across the federal government and others really to make this happen. So Cepheid's job was to sort of make the test, make the tool, but we needed all of this other collaboration to be able to ensure that the tool could be used. So the ITAP team here is in a big red box because they were just central and really important to this interaction that happened between Cepheid and the FDA and our work with NIH. We worked with CMS to ensure the test would be eligible for coverage and reimbursement. We worked with CDC and AASLD, and you'll hear from Nate and Andrew next around the guidelines that drive how clinicians and providers can use the test. And of course, we worked with our congressional partners around the elimination plan legislation. So we can go to the next slide, which is a timeline of kind of how things went. And this is not to scale and very, very abbreviated, but kind of on the left side, you sort of see where we started with this information and experience outside the US. A number of things happened early in the early 2020s. So there was the strategic plan that was released for hepatitis elimination. Importantly, the FDA downclassified HCV RNA tests, which allowed the opening of the regulatory pathway, also allowed for the test to potentially be CLIA waived and available in point of care settings. We had those early conversations with Dr. Collins and the White House team. And then in January 24, we launched the very beginning of the clinical trial for this test in the US. And as Eric said, at the end of June, we had an approval and authorization from the FDA, and we launched the product in July. We can click one more time. Really, that mindset and the Cepheid team in that period was we just had to shrink the timelines. Things that usually took a month or months became a week. Weeks became a day became a few hours. And as Eric said, we went through this with the mindset of there is no July. This was happening by the end of June. It took a ton of work. A lot of the Cepheid village was involved in this, many nights and weekends. And again, that collaboration with all of these teams across these partnerships was just really important for this project to move forward and for this to happen in the time frame that it did. So if we go to the next slide, this just shows with the workflow of the test. As Eric mentioned, we had this challenge with the collection device, and we had to essentially validate a collection device that was not approved in the US for use for molecular diagnostic testing for infectious disease, the microtainer. So the blood is collected, it's pipetted into the cartridge. This is a cartridge-based test. The cartridge goes into the instrument, and then the instrument runs the assay and provides a result. So it's a very simple workflow, but we did have to work collaboratively with all of these partners throughout this process to ensure that we had the right materials, that the instructions for use and the quick reference guides were appropriate for people to be able to run the test successfully in these decentralized settings. If we go to the next slide. So the top of the slide shows this sort of more traditional timeline for HCV diagnosis that Dr. Collins referred to. It could take days to weeks and multiple visits for a patient to get a diagnosis of HCV and then get linked to care and treatment. And at any one of those steps, any one of those arrows, a patient could sort of fall out and be lost to follow-up. So there's lots of opportunity for patients to be lost to follow-up. And what we have now in the red circle in the bottom of the slide is the ability to do a diagnostic test at the point of care where a patient can walk out of that visit or walk away from that site, knowing their HCV infection status, whether or not they have an active infection. And if they are infected, they can walk away either with a linkage directly to care or with medication in hand, so that we can get more people tested, more people treated, and more people cured and move towards that elimination goal in the US. If we go to the next slide, my thank you slide, there are many, many others that are not mentioned in this alphabet soup on this slide. This is not meant to be comprehensive at all. We had a huge Cepheid village, as I mentioned, working on this, that worked really, really hard. We worked with all of these people. I also want to call out the ITAP team. We spent many hours a day working closely with them. They worked really hard to get us answers, to escalate things as we needed. It was just an incredible, incredible collaborative set of work to make this happen. So I will end there and pass it back to you, Nate. Thank you.
Thanks, Jen. All right, so I'm going to take it over as a relay, and I'm going to talk more about shifting towards the implementation framework. Next slide. So just as a reminder, prior to this test coming to market, we were kind of limited in our HCV diagnostic approaches because everything ultimately ended up with needing to get that laboratory blood draw for a large platform test in a large laboratory for HCV RNA for diagnosis. Next slide. And a real big problem with that was that that entails incomplete testing, maybe only getting an antibody test, but not getting RNA testing. People getting lost to follow-up, a longer time for diagnosis. It also missed early HCV infections for those who were just recently infected and didn't have positive antibody at the time of testing. And then, of course, there was no point of care diagnosis. And this resulted in not a lot of people moving along that cascade of care towards cure. Next slide. So this was great. Like we got to this point and we celebrated, we did it. But then, next slide, there were just so many questions that came after that. Like, what does CDC guidance say about this point of care testing? Who would benefit most from this kind of testing? Where should we put these machines? Who should we test? Like, should I use the single step approach or should I kind of stick with the antibody first approach? And then what do I do with positive results? How do I get those folks treated? Next slide. So we sought about to answer a lot of these questions. One of the first questions was, what does CDC say about this? So here's a snapshot of our 2013 guidance on HCV testing and our algorithm. And here, there was nothing here that clearly or stated anything about single-step RNA testing or point-of-care testing. Next slide. But we went back and we dug through all the fine print of that guidance, just like, how do we find a way to greenlight this? Because we don't want to have to wait a year and a half for a guidelines development process to come up and tell us what we already know is rather evident. But we found it out. We looked at some of the fine text and the footnotes, and we saw that among those who had been exposed to HCV within the last 6 months, we had text that said, actually, you can go ahead and test those folks for HCV RNA because, again, that might be in that early infection period. And so we were able to quickly turn that around and get some language on our website saying very clearly that HCV RNA testing is currently recommended for the diagnosis of HCV infection among persons who might have been exposed to HCV within the last 6 months. And this is regardless of antibody testing. Next slide. Great. The next question was, what about the single-step versus two-step testing approach? A lot of benefits for the single-step one, but also a lot of benefits for the two-step approach, as you see in this nice image that Cepheid had developed. Next slide. And so we started thinking, all right, how do we create a framework for understanding what are the approaches we should use? So the first part, in the upper left corner was laboratory two-step antibody to RNA testing. That's what we've been dealing with. What we have has some benefits, certainly some downsides. Next slide, or next. Yep, thank you. But with this new test, it opens up an entire point of care testing option. So we could still do point of care antibody to point of care RNA testing. The benefits to that, obviously, it's all entirely point of care. You don't need any phlebotomy or laboratory access, and you can get all those results within the same encounter. A challenge here is that it still misses those early HCV infections, and then, of course, the added challenge of getting this a little bit more complex point of care test set up in some of the high impact settings. And it may be more expensive than laboratory approaches, depending, not always. Next slide, or next step. Yep. There was also this, lab-based single-step HCV RNA approach, which I guess has been around, but is technically off-label and isn't used that much. But next, advanced one. But really, there's this new single-step point-of-care HCV RNA approach. So similar to the point-of-care two-step approach, all that benefit of finger stick testing, with the added benefit of being able to detect early HCV infections. A challenging here might be if, depending on the prevalence of the population, that might end up being a more expensive process to get to your diagnoses. And then depending on the intensity of the volume that you're testing, you may run into some rate limiting steps. Next slide. So then we thought about who would benefit the most from point of care testing, and then where should we be setting up point of care testing capacity? And we boiled it down to a couple setting characteristics that we thought would be helpful for deciding what type of testing to do. The first is hepatitis C prevalence. If you're in a high prevalence setting, that might favor a single-step HCV RNA approach. For the encounter type, if you've got a brief encounter setting, such as like you're only going to be able to reach these folks within a limited period of time, that's going to favor a point of care test approach to get that result really quickly. Phlebotomy and lab access, obviously, if you don't have it, that's going to favor a point of care approach. But if you do have it, you can choose between the two. And then lastly, client volume. I think point of care HCV RNA testing can get really challenging if you're in very high intensity testing campaigns, just due to the nature of there's only so many modules within the machine to test at any given time. And then if it's a really low volume testing setting, you have to ask yourself if it's worth doing that investment in order to set up the machine and do all the testing for a smaller population. Next slide. So what we came up with was these potential testing strategies. So these aren't recommendations, these are considerations that you can think about, see if it's right for you. But in settings that had the highest prevalence, so these are places caring for like people who inject drugs. So that would be your syringe service programs, opioid treatment programs, or outpatient medication assisted treatments. Here, the prevalence of HCV is going to be pretty high. And so doing a viral first approach to ensure you're not missing any early HCV infections and that you're able to get all of that test results within the same encounter has some benefits. For additionally high prevalence settings, carceral places are a place we're particularly interested. Now here, the prevalence is not quite as high as some of these other settings, but high nonetheless, that depending on what part of the country in, you may want to consider a single step approach if you have a very high prevalence. For prisons, a lot of those places will have integrated infectious disease screening on intake already in place, so it may not make as much sense to add a point of care component to it. But for large jails where you've got a limited amount of time to diagnose someone, start them on treatment, we see a lot of potential benefit in these sort of settings. Next slide. And then for settings that have low to moderate prevalence settings, here maybe that two-step approach to testing might be more preferable. So that could be like in emergency departments or urgent care, STI clinics, primary care settings, remote settings, or maybe even pharmacies. Here, that two-step approach may be beneficial. And then depending on the nature of if that's a brief encounter, do you have access to laboratory and phlebotomy, that can help kind of shift you towards whether that's a laboratory or a point of care based approach. Next slide. So then the last question was, how do I pair point of care testing with treatment? And there are a lot of great models and pioneers in this. The Combi Clinic was a great example that Jen just gave. I think usually you kind of boil it down to maybe one of 4 different modalities. The first is co-located treatment. Go ahead and do one click. And that's a good strategy for ensuring that, you get that diagnosis, there's someone there to be able to then shepherd you into treatment. The challenge with that is that for these non-clinical settings where we need to scale up the most, you may not have the capacity to co-locate care. Go ahead, give it a click. Telehealth is sort of an emerging option. So places that maybe don't have the capacity to co-locate may be able to interface and get people on the computer or screen or telephone with a provider that can then prescribe hepatitis C treatment with point of care test results. Next slide, or next click. And then there's also the mobile unit example where we can take mobile units, set up everything we need as a one-stop shop, and either bring those testing and treatment capacities to sort of brick and mortar locations or even do community outreach. And then the last one is patient navigation. Here's where I don't really have a readily able ability to pair you, but maybe I can do some more warm handoffs, more aggressive patient follow-up, and linking to care. All of these, we want to make sure that we're not building bridges to nowhere. We want to ultimately get these people treated. Next slide. And so summing all that up, we're really excited about this new technology. We'd love to see it scale up in syringe service programs, large jails, opioid treatment programs, and mobile clinics in particular. And so I think this helps bring us one step closer to elimination. Next slide. And then this is just to say that this week, we published our hepatitis C point of care testing considerations document that details a lot of what I just covered that can be available for you all to read if you're curious and you want to think more about how might I implement this and if it's right for me. Next slide. And so with that, I want to pass the baton over to Dr. Andrew Aronsohn, who's an associate professor of medicine at the University of Chicago Medical Center. Dr. Aronsohn's a practicing hepatologist and a prolific researcher on viral hepatitis, and he serves as the co-chair for the AASLD IDSA HCV Guidance Committee. So he's going to talk to us a little bit about efforts underway to simplify hepatitis C treatment.
All right. So thanks so much for this invitation to speak. I'm really, really excited. And all these presentations leading up to this, hopefully I can shed a little light about what the HCV guidance can offer in support of this. Next slide, please. All right, I don't have any disclosures except I'm one of the co-chairs of the HCV guidance. So hcvguidance.org, hopefully many people on this webinar are familiar with this. And this is a document that is online that has all of our recommendations regarding hepatitis C care and treatment that you can find really in one place. We like to think of this as a one-stop shop. Next slide. So our guidance was actually started back in 2014, and just a little bit of history about this. It was really coming out of emergence of the DAA therapies. And what we realized is that these new medications were coming online really fast. And what were we going to do for providers who are on the front line of care? It wasn't really practical to wait a year or two for a journal article or guidance to come out. So what we did is the folks on the AASLD and IDSA teamed up for this combined document that we could really use together and create true real-time clinical guidance for providers. This was meant to be a living document that we would update with new evidence as it came along. And there is a publication every other year, but really this was supposed to be, something that you would refer to when you're seeing a patient in your clinic and you really want to know what the best treatments to use are. Next slide. Just very quickly, the governance. This is co-sponsored by the AASLD and the IDSA. We have co-chairs on each side and panel members representing hepatology and infectious disease. We also are fortunate to have community partners and members of the CDC who also help advise as far as our recommendations. Next slide. It's pretty popular. We have, last time I checked, had about 2 million individual users going to our site. And it's available everywhere because it's online, but we have documented usage in over 200 countries. But I will be clear, especially when I'm getting into some of the discussion around elimination, that since this is a US guidance, we're focused on treatment recommendations for the United States. So we use medical regimens that are available here in the United States. So it is US based, but certainly could many countries and folks in other countries have used this as a resource. Next slide. And here's just for those of you that are less familiar with our guidance, I just wanted to kind of show what it looks like. And what I really would like to hit home is what we hope is a hugely practical resource. So this is a very easily navigatable website online where you kind of hover over the characteristics of your patient regarding their treatment status, their genotype, things of that nature. And the first thing that will pop up is this very easy to read box with all the treatment recommendations, as well as duration of treatment. And then the evidence rating is written there. And as if you were to scroll down on this website, you would see all the clinical trials that have led up to lead to these recommendations. So you could, you can see the data yourself if you're interested. Next slide. And this is the groups of people for our treatment recommendation. Many of them used to be called special populations, but it's really just so good that they're not special anymore in the nicest possible way. And what I mean by that is these were difficult to treat and patients based on some of the comorbidities that they have. But now everybody is easy to treat. So when we're moving towards elimination and certainly thinking about new diagnostics, it only really makes sense if you can treat everybody. And I'm really, really excited to say that we can absolutely treat everyone, both treatment naive and treatment experienced, but also people with cirrhosis, including decompensated cirrhosis, those who are co-infected with HIV, acute hepatitis C, renal impairment. There's emerging data of treating people during pregnancy, as well as children down to the age of 3, and as well as people who have received transplantation, including with hepatitis C positive organs. We also have a newer section that outlines what you should do as a provider if someone has a treatment interruption, whether they've lost their medication or had trouble getting refilled or some other issue happened, and how to go about managing that patient that's had an interruption. Next slide, please. All right, so really what I want to get to is the importance of this point of care test. And what I hope to drive home in the next few minutes is really showing you what this means and what this is going to look like in the clinician's hands and how we are going to support this from the guidance to really incorporate point of care testing and guidance that many people use every day. So standard of care testing, and this has been shown in a few different ways through the past few presentations and different permutations, but standard of care testing has some drawbacks. So let's say a person comes in for testing, maybe in a provider's office, but maybe somewhere like a harm reduction center or an addiction center. And then let's say they have to wait for their test to come back positive. And then someone calls them and tells them to go to the hospital or a doctor's office or some other place where they can get treatment. And then sometimes even another confirmatory test is made. And then they have to wait to get treatment. And then they have to follow up after treatment. And as we know, and again, this has already been presented, each step of the way, you're going to lose patients. And when we're really serious about eliminating a disease, we can't afford to have those steps. So next click, please. So what we want to support, and I think that the whole point of why we're doing this webinar and all this fantastic effort that's gone into this is test and treat point of care. So what we are aiming to do and to help advise clinicians is test and treat, meaning a person comes in to a place, and that place could be a doctor's office, but definitely could be a van, or it could be a syringe exchange program, or could be a methadone clinic, where they offer the testing, and a point of care testing is key for this. And once that's deemed positive, then the medication potentially is waiting for them right there. So they're going to get the diagnosis, get counseling, get their evaluation, and leave with the medicine in their hand. And what you've done by doing this is eliminated about 5 or 6 steps where somebody, especially a vulnerable patient, can get lost to follow-up. Next slide, please. So where's the data to do this? Are we just crazy on the guidelines and we're just kind of making things up as we go along? Hopefully not. And this is the MinMon study, or what I call the keep it simple stupid study, but there's certainly nothing stupid about this. In fact, it's quite genius. So this is a large study that was published about 2 years ago, where they take a group of patients and did no pretreatment genotyping. They determined whether they had cirrhosis by using a FIB4 test. All treatment was provided at the time of entry, so when they first met them, which is exactly what I'm talking about. They had no scheduled treatment labs except for just 2 phone calls along the way. So they got 12 weeks of sofosbuvir and velpatasvir, and there was a phone call at week 4 and a phone call at week 22, and then they came in to have an SVR checked 3 months after they completed therapy. So really, really low touch here. You saw them once, and then you really saw them again when it was time to check to see if they've been cured. Next slide, please. So did this work? And it really not only worked, it was pretty phenomenal, the types of results we saw from the MinMon study. So overall, out of 399 patients treated altogether, so a relatively large study, 379, 95% were cured. This held true for people who are mono-infected with hepatitis C, as well as those that are co-infected with HIV. Next slide, please. But what about folks who have substance use disorder? So people with previous substance use disorder were cured. And even there was a small number, there was 12 patients, but 11 of 12 that had ongoing substance use were cured as well. And that really drives home that there should be no patients that are excluded from these test and treat protocols. Next click. And everyone asks, well, what happened to some of these patients? 3 people lost their medications. That's not so bad. And 2 patients discontinued. And I think that speaks to how safe and easy to take the DAAs are. Next slide. So we did something a few years ago where we simplified the HCV regimen. And these are what we call our one-pagers, which you can find on the initial landing page of our guidance. And what this is ideally there's a link to a PDF, and there's one for patients with cirrhosis, and there's one for patients who do not have cirrhosis. And without going through each step, this really takes you from who would be eligible for a simplified treatment, what tests you need to send in the workup, what are the regimens that are available to treat, what is the monitoring, and finally, what do you do when you're checking for an SVR, whether they're cured or not cured, and what needs to happen after that. So this was great, and I like to think of this as a first step, but there were some drawbacks to this, and that is that it actually requires at least 2 trips to the provider. This is really, since there's a lot of phlebotomy going on here, this is difficult to achieve in non-traditional settings. So it'd be really hard to do this outside of a doctor's office. And of course, now we have this great technology. It does not incorporate into a point of care testing. Next slide, please. So how is this going to fit into elimination? And when we thought about point of care testing and supporting elimination from the guidance, there's really 4 buckets, but I really want to hone in on this first bucket is we are going to be writing a new section, which should be published very soon. I'm going to get to the algorithm in just a moment about how to do test and treat, and this will be included in our guidance. So there'll be an actual algorithm utilizing this point of care test in our guidance. We don't have hepatitis B point of care testing yet, but there will certainly be updated when that's available. But a couple of other things, we do want to serve as a resource to optimize care delivery, especially amongst marginalized communities. So what kind of resources will be helpful for providers that are trying to incorporate microelimination in their own communities? Evidence that's going to desensitize centralized hepatitis C care, meaning what kind of evidence can we use and support by using other places to treat hepatitis C, and this gets into a lot of our non-traditional settings. And then finally, any new research going on in hepatitis C elimination strategies is going to be really important once it has data that supports it. And this gets into vaccines, long-acting injectable antivirals, and things of that nature. So next slide, please. But what I really wanted to finish off with is what our algorithm is going to look at. This meeting is about a week too early because we have approval for the AASLD, and we're just waiting on approval from IDSA to make this more make this publicly available, the actual algorithm itself. But what I am able to show you is what it's going to look like, and this is going to be available on our HCV guidance website, which will be our test and treat algorithm using point of care tests. So what will it look like? The first step, of course, is going to use the point of care hepatitis C viral load and other point of care tests, including HIV and pregnancy, that are already available. Then the recommendation is going to be to take a good history, where we'll ask pointed questions looking for decompensated cirrhosis, prior hepatitis C therapies, HCC, and of course, doing a good medication reconciliation. Then the next step will be a blood draw, but the caveat here is this is a blood draw where results will not be needed during that first visit. So an AST, ALT platelets will be needed to calculate a FIB4, and a blood draw to check for hepatitis B surface antigen if this point of care is not available. But again, this blood can be drawn, so meaning you don't need patients to wait for the interpretation of the blood draws or the results to come back. It just needs to be cooking. And then most importantly, the therapy is given during that initial appointment. Results will be followed up in the interim regarding hepatitis B and treatment if needed for that, as well as what happens if somebody is found to have cirrhosis by FIB4. And then an SVR check at the end of treatment, at 3 months after the end of treatment, to determine if somebody is cured, as well as further follow-up if they have hepatitis B or cirrhosis. So again, this is going to be very clearly displayed in an algorithm, so providers all over can use this and then adapt this to their own practices where they're taking care of patients. Next slide, please. All right, so that's all I have. I want to really thank everybody for their time and this amazing effort to get this test available. As a representative from HCV Guidance, we're happy to support Hep C elimination by simplifying treatment. You know, we can't do this without point of care testing. We're never even going to get close to being there. So we're absolutely thrilled that this is available. Stay tuned, because we will have a hepatitis C elimination section, but even more close to now and very, very soon, we're going to have our algorithm posted that's going to have exactly how to utilize point of care testing. That'll be free and available to all providers taking care of folks with hepatitis C. Thanks again for your time. I really appreciate it.
Thanks so much, Andrew. Now, if we could go to the synthesizing it together slides. So I think you've just all gotten a great peek behind the curtain about how the sausage gets made, and it's really fantastic because, as you can see, we've literally stacked all of these different processes in parallel to ensure that we can get across the finish line in as quick as possible. Normally, these would all be sequential and spread out over several years, and here we are probably a lot faster to where we would have been, but we still have a ways to go. So next slide. So as Dr. Aronsohn mentioned, we are really close to that truly same-day test and treat approach, and we really just need that hepatitis B surface antigen. There is some good news, which next slide you will see. The FDA has put out its proposal for down-classifying HBV diagnostics on the Federal Register. This is one of the penultimate steps needed to ultimately then down-classify HBV diagnostics, which has happened for HCV several years ago, which unlocked what you see today. And we hope that we'll follow a similar process. The comment period actually is still open and ends in November around Thanksgiving time. So if you want to give a plug for how important this is, feel free to follow the link below. Next slide. As mentioned before, we have this ongoing point of care hepatitis B surface antigen solicitation in anticipation that we're going to need this really soon. And so this process allows for one of the existing tests, or maybe a test we don't even know about, to come to market in a rapid fashion. That solicitation is open as well, and the link below is how you follow it to submit your application. Next slide. But, I just wanted to kind of level set and zoom out a little bit. There's still multiple barriers to hepatitis C treatment. Give it one click. So with all of the work that you saw today, we are taking out this two-step diagnostic testing barrier, but there's still all these barriers that exist. Next slide. And Dr. Collins had mentioned, what we really need to eliminate hepatitis C is what's contained in the National Hepatitis C Elimination Program, which would drastically increase funding for improving diagnoses, improving treatment, and then ensuring that there's a public health campaign for implementation to ensure that all of this is happening in the places where we need them the most, in the high impact and healthcare settings where the people with hepatitis C seek care. Next slide. And we're really hopeful an early analysis by the Congressional Budget Office suggested that for the Medicaid population, there's substantial cost savings. And should that be similar across the entire plan, that would be almost a sense of a green light. And so we're crossing our fingers for that. Next slide. So one thing that we wanted to pull in some voices is, although this is a new technology, there's already some folks jumping in and starting to use it, or maybe have been using it. So I wanted to invite to the floor Jennifer Havens first, out of University of Kentucky, to share very briefly about her experience using Expert HCV. Go ahead, Jennifer.
Thank you so much. I've been conducting a clinical trial, the Kentucky Viral Hepatitis Treatment Project, implemented in 2019. That's funded by the NIH. I've been working in an area in Appalachian, Kentucky, leading a longitudinal cohort study where we found high rates of HCV seropositivity amongst people who were injecting opioids. And so really when the folks from NIH came together to talk about what next steps were, really getting these new, at the time, new DAAs into the hands of people who were using drugs was a priority. So when we were looking at models of care, there weren't a lot in the United States. So we were really looking to other countries, and Australia has been mentioned. And they were already employing point-of-care RNA testing and same-day test and treat. So I made that the model of care for the Key Treat study in the hopes that when we got to the end, hopefully of the study, that we would have a point-of-care RNA test available and we would have results from that. So we've been using the Cepheid device. We were able to receive a research exemption from the company to utilize the assay, and we were using the quantitative test, but we've also used the qualitative test as well, since we are part of the clinical trial that was mentioned by Dr. Lai. And we were not only using it for diagnosis and that same day test and treat model, but we were also using it to determine cure. And throughout the study, when folks came in after they had initiated treatment, to look at adherence. But a really interesting finding from our standpoint was that it was a clinical trial. We did have loss to follow-up. And even though we've been doing cohort studies for years and have what we think are really fantastic follow-up protocols, we did have almost a quarter of folks who were lost to follow-up. But since we had been conducting RNA testing, we knew that almost 40% of the folks that we had lost to follow-up were actually, even though they hadn't completed treatment, were actually already undetected. So from a public health standpoint, I think that was really important. And having the point of care test allowed us to track that over time. So I think it's been a game changer, especially in very rural areas. The biggest take-home message, I think, for Key Treat for us was, given the lack of transportation in this area, there's no public transportation, no taxis, no rideshare services, no Uber, no Lyft. So really minimizing the points of contact was really important, I think, for this study. And the point of care test absolutely allowed us to do this. We also received a drug donation from Gilead so that we could put drug in hand to folks who tested RNA positive. And please do follow up with me if you have any questions about how we employed the point of care RNA test. I'm happy to answer those.
Thanks so much, Jennifer, for that brief note from the field. I want to turn it over to my colleague, Colleen Flanagan, who's from the New York State Department of Health.
Thanks, Nate. Hi, everybody. So we are excited for the FDA approval of the Cepheid device. We do have some state dollars to make an investment in the device. How we came to the decision as to where we are going to be employing the device is through our very large hepatitis C community-based rapid antibody testing program, where that program has been in existence for over 10 years now and has evolved over time in terms of how an RNA test was provided to an individual, from referral off-site to a small contract with Quest to do phlebotomy blood draws, which did not last very long because, as Nate pointed out, the sites that have the highest prevalence of Hep C do not have staff that can do venipuncture. And I think, as one of the other presenters pointed out, people who use drugs do not always have good veins. So the New York State Public Health Lab validated DBS, dried blood spot, for RNA testing, which was great. Finger stick, almost all of our sites are using DBS testing right now. But when we look at our data, we still see a fair number of individuals not coming back for that test result. And so that's really where we see a role for point of care testing, to be able to give that diagnosis at the point of care. And so our plan is to provide funding to our drug user health hubs, which are an extension to several of our syringe service programs in New York State. They offer wraparound services, including medication for opioid use disorder. And I'm happy to say some of them are actually offering hepatitis C treatment, some through some funding that we provide for co-location of hepatitis C in these drug user health hubs. And so we're excited to be able to see how this test works in these settings. We are concerned about workflow, and we know that that's something that these programs are going to need some technical assistance on. This is different. This is not a 20-minute test. And so what does that workflow look like? And then we also have to think about access to treatment. While some of these sites are providing treatment on site, the medication itself is not being provided on site. And so what does that mean? So appear to navigate that individual to the community pharmacy, establishing a relationship with the community pharmacy to make sure that that medication is there. Most do not stock it. And so those are some of the things that we anticipate as challenges. And I think, just thinking about what Jennifer said about the quarter of individuals who still did not return or were lost to follow-up, social determinants of health are extremely important, and addressing those social determinants of health as well. We are going to use the device in some of our clinical sites that have integrated hepatitis C treatment into primary care. I don't think most of these sites will be using the device in the clinic themselves, but more out in the community in high impact settings.
Thanks, Colleen, for that brief note from the field. And for a final brief note from the field, I wanted to turn it over to Tyler Bartholomew out of University of Miami.
Yeah, thank you, Nate. I'm going to be short because I know that we're running over time on this, and there's not much more I can add to this webinar. It's fantastic to hear this rapid translation of this technology and all the amazing people doing the work behind the scenes to make this happen. So I'll just highlight kind of two things that haven't already been discussed by Jennifer and Colleen. As we move forward and really focus on syringe services programs as these optimal venues to reaching people who inject drugs, we need to make sure we have equitable implementation of this technology. We're going to have a lot of programs in different places in the country that are going to have resources and their health department stepping in and fronting the bill to do that. But there are a lot of programs that don't have access to funding, support, implementation, help with getting this type of testing technology stood up. And so I would say focusing on equitable implementation needs to be key if we really want to make viral hepatitis elimination a priority and a thing. The second piece is integrating telehealth models into this type of care flow is really, really profound. My work and others at the University of Miami with the Idea Exchange program have been focusing on telehealth or peer-driven telehealth models, what we call teleharm reduction, to bring care to the people where they are. That might be at the needle exchange program. That might be in the field under a bridge. That might be on a mobile unit. There's different locations, different places as to which we can bring care. And what we see is that this type of telehealth approach of medication management, peer navigation, motivational interviewing, embedded into our harm reduction services, right? So not only are we able to drop off medication for folks, but we're also bringing them syringes, injection equipment, naloxone, fentanyl test strips, the whole comprehensive package of harm reduction supplies along with treatment. And what we see in our HIV treatment trials, thinking about that retention component is, even at 12-month follow-up in our intervention studies, we have 95% retention. And I think it's really, really important that we think about treatment models that are non-stigmatizing, low barrier as we move forward, and leveraging the SSP as an ideal and optimal venue to do implementation, but we need to make sure that those programs are supported, they're sustained, and that they can actually do these interventions moving forward. We have great progress, but if we don't, support the bed of foundation of how we're going to reach folks, we run the risk of not actually making a difference. So just really excited about the opportunity, but we have a long road ahead to make this a reality.
Thank you, Tyler. I wanted to turn over to John Ward. I realize we're at time, unfortunately. Go ahead, John.
Thank you, Nate. Thank you for bringing these speakers together. Thanks for the great overview of the ITAP process that led to the prompt licensure of gene expert testing for HCV. Felt like I was watching a little bit of a mix between 30 on ESPN and a Nova special to really go behind the scenes to see how that got done. So it was great to get that documented. And it really began to raise the really next steps in policy development and implementation. And really, in my mind, a real call for operational research that can help in these different scenarios that you raised as prime targets of where point-of-care testing for HCV can really be helpful. As we mentioned, this test has been available for a good number of years in other countries, and I think we can bring forward some of those lessons learned from other countries to inform providers here that are seeking to implement hepatitis C testing into their practice so that we can improve this gap and fall off between HCV testing and initiation of treatment. So I hope our future webinars can start revealing some of that operational research that can help implementers, help clinicians become testers and treaters of HCV with this new point of care test technology. I think, I don't know if we have, I know we're over time, so we're going to be closing off. Nate, do you want to make any other final remarks, or anyone else on the panel?
I think those were nice closing remarks.
I think that's all we have time for. Please keep in touch. Again, all of this content is available on globalhep.org in our webinar section. If you have any trouble at all finding it, just please contact me at jward@taskforce.org. And I want to thank my colleague, Lindsay Hebert, who was the producer and director of the webinar. Thank you all for your work to eliminate hepatitis C in the United States and by extension globally. Thank you very much.